Pictures of children in a swirling gray background. The UC Davis Mind Institute presents the Distinguished Lecturer Series. The Evolving Landscape of Autism Research, presented by Kelsey Martin.
I'm going to tell you a really brief history about sort of my path, and really with the perspective of what do I bring scientifically to the Simons Foundation in my own background. And then I'm going to talk about SFARI and sort of the 20-year history of SFARI with a peek into where we're going next.
Picture of brain cells over a light brown background.
So as David said, my lab is really interested. I am a cell biologist at heart, and I'm interested in the mechanisms by which experience changes connectivity in the brain to store long-term memories.
Picture of a neuron in the brain and in culture.
We know that long-term memories require new gene expression, and also that they can be synapse-specific, or at least subsets of synapses can undergo long-lasting changes in structure and function.
Picture of a neuron with multiple dendrites.
And so as a cell biologist, that raises two really interesting questions. And they're derived from the fact that neurons have this incredible, beautiful morphology with long, elaborate processes, compartmentalization with up to thousands of synapses. And so my lab has been interested in understanding how you can regulate gene expression, what the mechanisms are. A lot is known in cells like lymphocytes, but they're easy because the distance between the plasma membrane and the nucleus isn't that far. But in neurons, we've been interested in understanding how does a signal get from a really distal synapse back to the cell body into the nucleus to regulate transcription. But also then, if you can have sort of synapse or branch-specific forms of plasticity, how do you spatially regulate that gene expression at the level of those subcellular compartments? And so those are the two questions we focus on, and we, as David said, we work in mouse hippocampus. We also work in sensory motor neurons from aplysia. We actually no longer do for a variety of reasons. Aplysia have wonderful advantages for cell biological studies. But they've sort of been overcome by all of the tools that are available in mouse. And there's actually another really interesting feature. It was great for me to move to Los Angeles because Aplysia californica lived in the ocean. And so I had a scuba diver who would collect them for me. And it became more and more difficult to get healthy animals. And I really think it's with global warming and the water temperatures, they had to go further and further out, couldn't get them. But
pictures of cytoplastic isoforms and dendrites in different colors.
So my lab, in the first question, we've found roles for localization of RNAs, for translation of those RNAs at synapses. And the second question, we focused on how do soluble signaling molecules travel that long distance from the synapse back to the nucleus, and what is the impact on transcription. In my scientific career, I've done one project that was related to autism, and so I'm going to tell you a little bit about that one. So that was really my first foray into something that was relevant to autism, and it was looking at an RNA binding protein called RBFOX1, and we looked at it. It is a SFARI SCORE2 gene. That's not why we studied it. We studied it because it itself is spliced into two forms, one that's nuclear, one that's cytoplasmic, and it binds to a really well-defined sequence on the RNAs. And as I said, we're interested in how RNAs get localized, how their translation is regulated. And so we did experiments where we knocked RBFOX1 out in hippocampal neurons and then re-expressed either the cytoplasmic isoform, so you can see it's in the somatic cytoplasm, it's out in dendrites that are labeled in the cyan with MAP2, or the nuclear isoform, you can see that's all in the nucleus. And then we did RNA sequencing to see how the cytoplasmic isoform changed gene expression or how the nuclear isoform changed gene expression. And what we found is that they regulated the expression of very different sets of genes, and that the cytoplasmic isoform regulated the level of expression of genes, whereas the nuclear isoform regulated splicing and the binding. And then we did something called iClip, which is a form of looking at the targets, doing immunoprecipitation from that RNA binding protein and looking directly at those targets. And we found that the sites that it bound by the cytoplasmic isoform were all in the 3' UTR, whereas the sites that were bound by the nuclear isoform were all in introns, consistent with those functions.
Graphic of cytoplastic mRNA stability and transformation.
We went on then, and working with my colleague at UCLA, Dan Geschwin, did experiments to identify what were the targets, and we found that the targets of the cytoplasmic isoform are really enriched in SFARI genes and in autism genes. I have to say, of course, I wrote grants to SFARI. I never got any grants from SFARI, but that was, about as close as I got. But it was a really nice cell biological story where we could show, again, that nuclear isoform is involved in splicing. And we went on to show that the cytoplasmic, we could identify the targets, and we found that what that binding in the 3' UTR does is it's either adjacent or overlapping with microRNA binding sites, and so it stabilizes those RNAs when it binds and actually promotes translation of those RNAs. So we're able to identify a function for that. So that really was sort of my foray into autism research. But I have, I'm an MD PhD. I've been really committed to trying to make sure that clinical medicine and basic research benefit from each other in both directions. And as David said, that really led me to become the dean of David Geffen School of Medicine, in part because I was very committed to making sure that synergy was realized. And I have to be honest, for me, a lot of it was in neuroscience because I feel like we know so little about the underpinnings of many neuropsychiatric disorders that there's really a great need. So I became dean of the School of Medicine for 6 years, and then I got a call from Huda Zogby, and she said, hey, would you maybe be interested in this position? I think you might be interested in the role. They're looking for somebody to run SFARI, and it was not something that I had thought about previously, but it just, I realized after I talked with her and I got off the phone that I was very excited about what could be done, and I was excited, ironically, in a way that I felt that it was actually quite consistent with what I was trying to do as dean. I would be completely humble and say trying to do and maybe not completely able to do, but I realized that going here, it was going to a foundation that was completely committed to basic science, but particular in the work in autism, was that basic science with the purpose of making a difference in people's lives.
Picture of the David Geffen School of Medicine over a map of the U.S. with an arrow pointing to another picture of the outside of the Simons Foundation building.
And so, somewhat to my surprise, and also I was old enough, my kids were out of the house, it felt like if I make a change, I should make it now. So I moved in 2021, physically moved to New York City, although I do still have a small lab on active recall at UCLA, to the Simons Foundation.
Picture of Jim and Marilyn Simons.
I just want to, give you sort of an overview of the foundation. It was founded in 1994 by Jim and Marilyn Simons, and it's a philanthropic organization that has what I consider to be a very noble mission of advancing the frontiers of research in mathematics and in basic science. And it does it both by grant making, there's an in-house research program, and in public engagement. So a big arm that's looking at how do we ensure that the public is engaged and interested in science. Jim was an incredibly renowned mathematician who then started Renaissance Technologies, a quantitative hedge fund. And again, because of their commitment to basic science, they started the foundation to be able to support research. And the foundation has 2 parts. It's literally in 2 different buildings for people who have been there. And one is largely the grant-making division. And when I came, they put together the autism program, which is the home of SFARI, with the neuroscience collaborations. There's a math and physical sciences division, a life sciences division that's now focused on ecology and evolutionary biology, and then Science, Society, and Culture, which is a grant-making division focused on public engagement. That's the home of e-publications quanta, the transmitter. For those of you who know that we have a documentary film studio called Sandbox Films that makes science documentaries, so really trying to do that outreach, and also providing grants throughout the United States to support programs that bring people from all different ages to think about math and science. And then there's an intramural program that has 5 computational centers, so it's all dry lab, 1 in astrophysics, 1 in biology, which is largely biophysics and genomics, in mathematics and neuroscience and in quantum physics, and that's supported by a really robust scientific computing core with really outstanding compute infrastructure that also benefits the grant-making division. So we also, we have an incredible neuroscience ecosystem at the foundation. So the division that I oversee in autism and neuroscience, society and culture, the transmitter, and then both the Center for Computational Neuroscience and the Computational Biology, there's lots of synergy between all of these, and we meet very frequently. So the missions of each of the programs within autism and neuroscience includes SFARI, where our mission is to advance the basic science of autism and related neurodevelopmental disorders. And I'll note that we changed that mission about 2 years ago. It was really focused on understanding diagnosis and treatment. And as we took time to really think about where do we fit, both within the foundation, the Simons Foundation, but also within the broader funding ecosystem, and being aware of other funders coming online, especially ARIA, that's run through the Coalition for Aligning Science with funding from the Brin Family Foundation. We felt like our sweet spot is to really continue to support that basic science and to partner with other funders. And then the neuroscience collaboration has a different mission of supporting neuroscientists to, convening them to address fundamental questions about brain, not disease-focused. I tried to blur the lines there because we actually are increasingly, and I'll try to show that, building bridges between those activities. The informatics unit has a mission of operationalizing the foundation's commitment to open science, and I think that's another thing that I feel very lucky. I wasn't there when this was set up, but that from the very beginning, SFARI had this commitment, as I'll show, to making sure that all of the data that's generated through SFARI-funded activities is made available to qualified researchers. And then, of course, we have a strong commitment to early career programs. I think this is more important than it's ever been to making sure that the next generation of scientists thrive, even in this current moment. So SFARI, just a brief history, SFARI was launched in 2006. The founding director was Jerry Fishback. And so he was the director of SFARI from 2006 to 2013, and he actually then moved and started the neuroscience collaborations. And Louis Reichart came as the director of SFARI. We have, currently we support over about 300 investigators, and since the launch, it's actually hard to track all this, but it's at least 750 investigators. So really robust community of autism researchers. And the annual budget across the division is about $155 million, so substantial budget to make a difference in the research that's being done. Not substantial enough to make a difference for the NIH, but anyway. And we have, I think, about having 3 buckets of activity, one in convening, so we host a lot of workshops and meetings. I've seen many of you at our workshops and meetings. Grant making, we provide grants to individual investigators, collaborative grants, early career grants, and we run 2 institutional programs, one at MIT, which was Jim Simons' alma mater on Simons Center for the Social Brain, and one at University of Edinburgh called the Simons Sidbey Initiative for the Developing Brain. And then I think a really important part is building of resources. And here it's biorepositories, cohorts, data, and that's really intended to, again, amplify the impact of all the work that SFARI supports. I do want to just say a word about the change in our mission. because I've actually been a little bit surprised at how many people will come and say, oh, you don't fund human research anymore, or you don't, and nothing could be further from the truth. And so for us, what does the commitment to basic science mean? It really means looking at what are the mechanisms of disease. For human condition, you have to understand that in human populations, and then you can dig down in other systems. But we recognize that that's so, it really is, and I keep meaning that now. It's been 3 times I've shown the pyramid, and I've said I need to have a circle because it's not a pyramid, but it really is that communication between the basic science and translational research that builds on those basic understandings, and then the clinical research that takes those translational insights and puts them into human populations. And what bridges all of those, I think, besides communication, are data and resources. And so we've put a lot of effort into making sure that our data and resources are very strong. And towards that end, a new head of informatics, Eric Wong, with a lot of investment in making sure that our data platforms are rich and accessible. And SDBR, which is a SFARI data and biospecimen repository, a director, Lauren Bauer, of that, and thinking of that team is really the navigators for researchers who want to use our data, our biospecimens, so making sure that that's as accessible as possible. When I came to SFARI, Jim Simons sent me this article, which is from the Science Philanthropy Alliance, and he gave it to me because it was a really beautiful history of how they started SFARI and what they thought about. And it described, David Amaral and I talked about this briefly last night, but it described hosting a meeting and having a roundtable of really distinguished neuroscientists, most not working in autism, and just coming with the question of, what should we do to make a difference? Where should we invest our resources? And the 2 ideas that really came out of that I think sort of have stimulated a lot of the activity over the last 20 years is, one, that they should raise the quality of talent. They should bring a lot of people into the field. And I think that fact that now there are 300 investigators who are supported by SFARI, I think it is a very robust, rich research field.
Picture of 2 identical twins, fraternal twins, and siblings.
And the other was that genetics would be a fertile ground for research. And that really was because of the finding of the high heritability of, it says most, but it isn't, I meant to change that. This is an older one. It is a highly heritable condition. And if for identical monozygotic twins, if one has autism, the likelihood of the other being about 70 to 90%, for fraternal twins closer to 30, and for siblings about 20. So that tells you, there's a genetic component, but it's not just genetics. But the fact is that at that time in the history, at the early 2000s, the tools that were coming online for gene discovery, it's sort of a ripe moment for using those tools to better understand autism. And that's really what motivated the first cohort that SFARI created, invested in, and that's the Simons Simplex collection. And the features of the SSC, or Simons Simplex collection, that I think made it so impactful are, one, that commitment to open science. And again, I have to keep reminding myself, this is like 2005 when this is being designed. That's impressive. A simplex design, so a design with one proband in a family, unaffected sibling, unaffected parents, to be able to look specifically for de novo mutations. So that was the approach. A really standardized data and biospecimens, so 12 sites around the country where Kathy Lord went around, I think with Marion Greenup, and made sure that everybody was using exactly the same measures. And unbiased genomic analysis. And I think those principles have been really successful. And it's, we continue to get lots of requests for data from SSC. And I think that many discoveries have been made from that cohort.
Picture of the U.S. map with red markers showing the locations who participated in the study. List and graph of the many genes associated with autism.
The payoff now is that that hundreds of genes have been identified that are associated with autism. I often do think, and in conversations with Jerry Fishback, there's this, I sense this thought in early 2000s that understanding the genetics would help address the heterogeneity, but it clearly has not addressed the heterogeneity, so I think that's important to recognize. And that, when you look at this is a list of the SPARC genes in some of our cohorts. This is from Thomas Bourgeron's lab. And the reason I like this particular, there are many examples of this type of a graph, is that it shows there are 13,000 individuals they've looked at. These are the number of carriers who have any particular genetic variant. This is the odds ratio. So very high odds ratio for some of these CHD8, CN2A, SCN2A. But, relatively small numbers of individuals who carry them. So it tells you that this accounts for, a substantial, but still a fraction of individuals with autism. But we are still totally committed to supporting cohorts. And, well, again, as I said, SSC, it's now closed. It ended in 2013. It had 2,600 families. We still get lots of requests for data, for biospecimens, for other studies using SSC. The Autism Inpatient Collection is a cohort that has about 1,500 participants. It was co-funded with Nancy Lurie Marks Foundation. And I just have to make a note that that's another commitment that I have, and I love being at a foundation. Academic schools, as much as important as collaboration is, there's always competition between even UCs, I would say. And so we're really committed to making sure that we partner with other funders to make sure that there's non-duplication of efforts. So this is work with Nancy Lurie Marks Foundation. Matt Siegel was the PI. This is 6 inpatient sites, children hospitalized with autism, and we released last year. All of the data is available on SFARI base that has genetics and phenotypic clinical information. And the 2 ongoing cohorts include SPARC, and that the PI of SPARC is Wendy Chung at Boston Children's Hospital. We currently have over 445,000 participants, and there are over 190,000 with autism. That's a phenotype first cohort, so individuals who have a professional diagnosis of autism can join. They can, they're invited to consent to provide a saliva sample. We had been doing whole exome sequencing. This year we moved to whole genome sequencing, and we will this summer be releasing 50,000 whole genome sequences, so a really large number more by the end of the year. And then Simons Searchlight, which was set up as a genes first, so individuals who have variants in those many genes that are associated with autism can join Simons Searchlight. We currently have over 7,200. Those are actually carriers and other family members in 184 different groups. So it's you can see how that starts to spread in pretty large ways. And again, all this data is available on SFARI base to qualified researchers.
Graphics showing the steps that the Simons Foundation goes through with a participant in their study.
I also just want to highlight the Research Match program, which is a way of connecting families, participants in the cohorts with other research studies. So what happens is individuals send their study in that they would like to recruit participants from SPARC to. That's reviewed by a participant access committee, by a scientific review committee. If it's approved, then we inform participants who are eligible to participate in that study. If they consent to join in, they then participate in that study. The study data can lead to new findings. We then can take that data and put it back into SFARI base.
Graphics showing the 4 steps for getting genetic information to Simons Searchlight.
And there is a nice, feeding between these, because when you recruit family members and you collect data, if there is, we do return of genetic results. So about 8% of participants get a genetic result returned. And they're also invited to then join Searchlight. And then we can then again match participants to research studies. And I would say, one thing I'm really pleased about is It used to take, and for anybody who's ever involved with families who in Spark, I think this is important, it could take years for people to get their result from there. And so we've now changed the way that we're doing our sequencing, and the results all are returned within 3 to 5 months, which I think is really important for family members.
Pictures of the 4 boards and advisory committees for the Simons Foundation.
I want to just make a note that, that I talk about our mission is basic science, but the cohorts are really about how do you engage the community in a study, and so there's a huge effort. For example, this is just showing for Spark that we have a scientific advisory board, we have an outreach and engagement advisory board, we have a community advisory council, and then this participant access committee, which is the committee that looks at the projects that are being submitted for research match. Why is this important? It's because we can't do this without making sure that the work that we're doing is going to be important to the participants, and so we get that feedback from them throughout the entire process.
Graphics showing information starting from proteins to cell biology to cell types, then to neural circuits and to cognition.
I think that there's a kind of a recipe that I'm aware of, where you identify a gene, you make a mouse model, and then you start to understand some of the underlying biology that you can then use to understand some of the human condition. And that has been a strategy at SFARI.
Picture of a mouse, rat, zebrafish, and pluripotent stem cells and neurons.
To support that, we've supported the creation of mouse models, of rat models. I see at least one person who's in our Autism Rat Consortium, of zebrafish models. We create a repository of patient-derived induced pluripotent stem cells from Searchlight participants. And here, I'll just make a note that we will be, within the next probably month, releasing something called a request for nominations, where we're going to have, the way we're going to decide which iPSCs we're going to make is we're going to have researchers come to us and say, I'd like to have this particular iPSC line made, why, and we'll use that to drive the ones that we're making going forward. And that's also a wonderful collaboration with Nancy Lurie Marks Foundation. And then, of course, Autism BrainNet, Postmortem Brain Bank, gold standard for trying to understand human brain conditions. But, so how that has been a lot of the funding, and I think I do want to just point out some of the...
Picture of Lauren and David. Picture of a human body showing the central nervous system and the peripheral nervous system. Picture of hairy skin and glabrous skin.
Things that important kind of concepts that have been learned from these animal studies. So I'm just going to talk about 2 studies very briefly. One of them is from Lauren Orefice and David Ginty, funded by SFARI. Both study, David Ginty has really pioneered the study of the neurobiology of the sense of touch, looking at hairy skin and glabrous skin. And of course, they realized that sensitivity to touch is a common feature of autism. And they thought, well, this is what we study. Again, this is a group of people who hadn't looked at autism. So I think it also gets to that point of bringing in different talent. What they did was they took a set of genetic variants that are linked to autism, including MECP2, the GABBR3, I think FMR, if I remember correctly, and they...
Picture of the abstract of the study showing mice being tested.
Did experiments in which they showed that if they just knocked those out in the peripheral nervous system, that they show that in tactile sensitivity. But not only that, if they do that in juvenile animals, they also develop anxiety and social deficits in behavior. And that there's sort of a developmental time period for that happening. So really interesting biology about how the peripheral phenotypes impact central phenotypes.
Graph showing tactile sensitivity of mice decreasing after treatment.
And then they went on to show that they could use a compound that doesn't cross the blood-brain barrier and show that that could rescue many of these phenotypes. And so you can see in their data, for example, and this compound is guvacine, and they look, and these are different. I realize I didn't put look up the names, the legend here, but these are different mouse models, and after this injection of this peripherally restricted a compound, they could completely normalize it, and they then worked together with Deerfield to create small molecules that would act in that similar way. And the truth is that none of these have worked. But the fact is, I still think it's a promising finding, and I think it hasn't worked for a variety of reasons that are more about sort of pharmacokinetics and toxicity. And so I think that there's further chemistry to be done there. And then the other, of course, is that when you identify these genetic variants, that then opens the door for those individuals for which it's possible for gene therapies. And so this is just a list, for example, in Angelman's and Rett's, SCN2A, SHANK3. But there's some really basic biology that I think has been really important.
Graph showing reversal of neurological defects in a mouse model.
And so, for example, studies from Adrian Bird, partly funded also by SFARI, and that's part of that University of Edinburgh investment, showing that in a mouse model of Rett syndrome that you can reverse by providing a mini gene for MECP2, and you can reverse it at later not every phenotype, but you can reverse it later in development. And so I think that those findings really open the door to thinking about critical periods for intervention. And so, these are kind of, I think of them as, it's hard to translate from a mouse to a human, but I think some of these principles that you can learn really do open windows to thinking about how one can have human interventions.
Pictures of brain cells and a modeling of connection between genes in the brain.
I just had to say, because I'm here with David Amaral, something about Autism BrainNet, so I think that's also another incredible resource for us. And it's really this, and Cindy, repository of autism brain banks, now over 400 brains that are available. And just highlighting, it's an opportunity to make, again, incredible discoveries, whether it's about loss of Purkinje cells in cerebellum, whether it's about changes in gene expression in synaptic genes or immune genes. I think that, I look at that and I think about emerging technologies, just as there were emerging technologies in gene discovery 20 years ago, things like spatial transcriptomics, single-cell, RNA sequencing, proteomics. They're just incredible opportunities for the future.
Graphics showing information starting from proteins to cell biology to cell types, then to neural circuits and to cognition.
But I have to say, I told you I'm a cell biologist, but when I look at this field and I think, well, what are the same kinds of opportunities that were available 20 years in gene discovery, I can't help but think about the opportunities in really parsing heterogeneity at the behavioral level and really understanding that cognitive and behavioral aspect to understand heterogeneity. And, really being able to move away from measures that are more subjective, that are episodic, that are more distant from the core biology, to going to really more objective, continuous and longitudinal as a neurodevelopmental disorder, having that. And this, of course, here I am where you have the Autism Phenome Project, so I think I may be speaking to the choir here. That are more passive, that are real world, that you can do at home in some cases, and that are closer to the core biology. And because we have these very large cohorts, we're interested in measures that are scalable. And so the foundations, SFARI has hosted RFAs like the Human Cognitive and Behavioral Science, cross-species studies, where we funded individuals to do different projects or some small cohorts. So here's one example of another similar type project that we supported, that Elon Dinstein did he was on sabbatical at SFARI and set up a project called the Simons Sleep Project, where we sent 3 different devices to 100 families in Spark who had one teenager with autism and a sibling without autism. It included a smartwatch, EEG headband, and a sleep pad that measures pressure from the bed, a cell phone that all the data would stream to and then stream back to the Simons Foundation. We had patient questionnaires. And all of that data is now available, and I think that that's how you get to it, the QR code at SFARI base. This is a publication that came out that describes the resource. You know, the findings from it were that there's, not surprising, greater sleep onset latency in autism. There were interesting findings about parents thought that their kids were asleep a lot more than they actually were when you look at the objective measures. But it's, you know, it's a small cohort, very heterogeneous. The data is being requested a lot, which I'm very satisfied by, and I think a lot of it is to try to build pipelines for analyzing this data.
Picture of a child in a kitchen holding up her hands and body image showing her movement.
We have, looking forward, one of the things that we're very interested in is how do we use video and audio-video data to parse that heterogeneity, and combining that with AI machine learning methods for analyzing that data. So in January, we hosted a workshop that was really wonderful. It included SFARI-funded investigators. It included investigators from our Simons collaboration on the global brain, from other areas. So people who are working on how do you understand non-human animal behavior using all sorts of different methods that allow you to identify, sort of measure the syllables of behavior in animals. And then we brought in people who are working on, gait analysis in adults with neurodegenerative disorders, people working in computer vision, and had them work together, and it was incredibly exciting. And part of that is also driven by a project that was done as a collaboration between Olga Troinskaya, who is a faculty at Princeton, but also a member of our in-house research program in the Center for Computational Biology, who's a computational biologist and genomicist. And she undertook what's called a person-centered approach, so trying to take a generative mixture modeling approach, looking at SPARC data to begin with, and to see if, her original question is, if we do this, can we try to get a better understanding of the underlying genetic architectures? This paper got a lot of attention, so probably a lot of you have either read about the paper or read the paper.
Picture of genomic data and phenotypic data from the SPARC study.
But really briefly, they used SPARC, where they looked at the genomic data, but they also looked at the phenotypic data that we have that honestly is fairly shallow, fairly incomplete, and working together with clinical psychologists, identified a number of questions that were available in about 5,000 participants where we also had genetic information. And using that, it was a little over 5,000 participants, where they took data from the social communication questionnaire, repetitive behavior scale, child behavior checklist, and background history, and were able to pull out 4 really distinct subtypes. They're not very surprising. I don't think that they're the only subtypes that would exist, but they were distinct and together in what they called the moderate ASD with developmental delay, social behavioral, broadly affected, and moderately challenges. And what was amazing is they also then went back to SSC, used the same protocol, replicated the exact same 4 subtypes. And when they then looked at the biology, they found the underlying genetics, they found that different types of genetic variants occurred in different populations. So for example, in the broadly affected, included primarily de novo mutations, whereas the moderate challenges was more common variants. And interestingly, when they looked, they actually impacted different biological processes. So you can see moderate challenges was largely histones, broadly affected synapses, social behavioral, again, chromatin and mixed ASD with DD, cell neuronal excitability. When they looked at when these genes were expressed using post-mortem brain tissue, they saw that there were different developmental subtypes. So for example, the social and behavioral was later onset of gene expression. So this is, to us, was an incredibly promising approach. If you can do this using snapshot, pretty minimal data, then what can you do when you have a much richer source of phenotypic data? And that they're not the only paper that has come out like this. There's also a paper from Varun Warrior, from a preprint from Thomas Bourgeron, another from Eric Morrow, using sort of similar AI ML approaches to looking at data and being able to really parse differences in different phenotypes. So that has encouraged us going forward to make sure that we're going to be supporting this kind of research in the future. And then we launched an RFA for letters of intent that we're calling the New Ideas RFA. And part of that is inspired by the fact that when Jim and Marilyn started SFARI 20 years ago, it was really to bring in new ideas. And again, we think that there are a lot of new ideas that are out there and for which there maybe hasn't been an avenue. And so this is a call, and it's an RFA, but it's also important. Oh, keep eyes open, but it actually, oh yeah. So it's actually there with a perspective that explains like what we're looking for, but it's a rolling submission with the idea that we'll get back in a relatively timely manner. I do want to say that keep your eyes open for the geneticists here. I'm talking about other approaches, but we're working on a new genomics RFA, and so there again, lots of opportunity in genomics. I just said we're launching we're going to be releasing a very large number of whole genome sequences, so all of the discovery that can be done looking at the non-coding sequence of the genome, lots of interest in looking at variants of unknown significance using different functional assays to see if we can better understand those. Interest in in what population should we be using long-read whole genome sequencing, either to look for epigenetic changes or to look for structural changes. So that should be released within a few months, because I do think that it's important for us to continue that SFARI investment in the genetic architecture. So I'm going to just quickly switch to looking at neuroscience collaborations. Again, I talked about those early career programs, and I want to make sure that everybody's aware of the Fellows to Faculty program. We're actually, I think, doing all of our interviews next week. So this is a program that's meant to support it's not just mentored positions, so you can only be a postdoc for most places 4 or 5 years. Some people are longer, and then they become instructors, but people who are in that mentored position, ready to apply for a job, and we provide support for up to 2 years, and then 3 years of their independent position. And we do that in the areas that we support, so including in SFARI as well. So that will open again in fall of 2026. And then we have a program called the Shenoy Undergraduate Research Fellowship in Neuroscience, named after Krishna Shenoy from Stanford, who was one of our leads in one of our neuroscience collaboration. And the goal really is to fund undergraduates who have had limited access to research for a variety of reasons. They might be at a community college that doesn't have research labs. They might have to work for pay to be able to support themselves. And so we provide salary support throughout the academic year, and if they want to stay during the summer, to work in the lab of a Simons Foundation supported lab. So, and those applications open in December. So briefly, the neuroscience collaborations, there are currently 3. There's one in the global brain that actually was led by David Tank. The collaborations are a little different. They're 10 years in duration. And that's a vision that it's long enough to do something really important, but you're able to then evolve into a new project. So Global Brain, started by David Tank, is focused on neural coding and dynamics. Again, I think really important back to 2014. This is a project that brought theorists together with experimentalists to work collaboratively on projects. It was a big collaboration. You can see there are about 100, you don't have to count them, but there are about 100 investigators. It ended, it had an extra year because of the pandemic, but we're continuing to have meetings. Last week we had an annual meeting with them, and because of those fellows to faculty, there's another 2 years where we'll keep that going. Plasticity in the Aging Brain is led by Colleen Murphy. It's a bit smaller. It's about 30 individuals. It's focused on healthy cognitive aging, includes working across species from killifish and C. elegans to humans. And it's organized as smaller groups of individuals working on different projects: blood-borne factors, navigation, transcriptional changes with aging. And the newest collaboration, the Simons Collaboration Ecological Neuroscience, was launched in July. That's led by Mate Laniel from Cambridge, and it's focused on the idea of affordances, how the brain encodes opportunities for action in the world. It actually does include one SFARI investigator in that. I think it is relevant to autism research. It was launched after a process that we did when we realized that the global brain was ending. Rather than take a top-down approach and say, we want to study this, we made a call for people to submit vision statements about something they could do that was very bold, that would take 10 years, that at that time would not be funded by the NIH or other traditional funding sources, and we got 250 amazing vision statements. It took us much longer to review them than I anticipated. It was really inspiring, a little depressing because we couldn't fund all of them. This is one that we selected from that group. And there are two others that we're working on with the groups that submitted them.
Graphics showing the intersection of theory, genes, activity and evolution, and a graphic picture of a brain inside a body.
One in cortical development, and it really looks at the hypothesis that cortical development is governed by mechanistic rules that are a result of genetic programs and activity-dependent processes and evolutionary innovations. And so that, we've held several workshops. We're working to shape that. And one of the reasons to do this for me is I really felt like here's an opportunity to take that basic neuroscience and put it in a way that's really synergistic with the goals of SFARI. The other that we have a workshop next week with that collaboration is on brain energetics, and that's really testing the hypothesis that the generation, supply, and utilization of energy places limits on the architecture and the function of the brain. And again, it's because this is a moment where tools are available for us to understand this. And it's exciting because it brings together very different people working from scales of, into synapses to human brain imaging. And we're really excited to keep building these collaborations. So I talked about the work with Olga Troyanskaya, with the Simons, with SFARI. She's also working with us on that plasticity in the aging brain, because when you have to work across all those different species, it's really hard to know what age of a killifish is the same as the age of a human being. And so she's using computational methods to look at gene expression, to look at other aspects of biology to see if we can address that question. And then we've started some work with the Center for Computational Neuroscience that's led by Eero Simoncelli, where we decided we were going to try to do a collaboration that was not just giving money to people, but having that collaboration embedded in the computational wealth of information at the foundation. And so we've held a workshop on the idea of canonical cortical computation, and the idea is we are building a collaboration that will include experimentalists out at universities, but working in collaboration with the theorists and the computational scientists in the Flatiron. So a different model for how we put those two different parts together. All right, so just to summarize, really, if I think about what are the guiding principles for SFARI, it's really the understanding that genomic architecture does give you a rigorous way to understand autism, biology, and heterogeneity, but that quantitative and longitudinal phenotypic information is critical to parsing that heterogeneity and to elucidating its underlying biology, and that we can use that framework to really begin to look at the underlying neurobiological mechanisms, again, really taking advantage of that partnership with the neuroscience collaborations, and then increasingly with the computational expertise that lives in Flatiron. And our belief is that investing in that basic understanding is important to build the knowledge base that's needed to do more translational work and then to do more clinical work. And I put that because I just think it's so important for basic scientists to really understand what it means to work with the community. So all of that work is really community participatory studies. The importance of bringing new talent and scientific ideas into the field of autism research. They certainly did that when they asked me to run the program. And then a deep commitment to open science and to data sharing. And I just want to end by acknowledging, I mean, one of the nice features is that there are a lot of people at Simons Foundation who've been there from the very beginning. I keep hearing, I'm employee number 6, I'm employee number 3, they've really, so we have a lot of people, John Spira, who actually, Monday was his last day, so he retired after 19 years. Marta Benedetti, Alan Packer, Julia Summer have been there for 15 to 18 years, 19 years. Britta Gunderson, Paul Wong, and then newer individuals, Amy Norovich, who is joint between SFARI and neuroscience, Jen Foss-Feig, and then informatics, Aaron Wong, SDBR, Lauren Bauer, and Jennifer Valdivia Spino runs all of our early career programs. And then in neuroscience, Alyssa Bikini-Schaefer, Sachin Ranade, Hannah Bayer, and then I have a very small but mighty team at UCLA of 2 research scientists, Jenny Akiro, Sylvia Newman, and Marika Watanabe. And I'm happy to answer any questions.
So just to let you know, it was a great presentation. Most of it was over my head.
I'm sorry.
I'm a parent,
Okay,
but
that's
I
okay.
appreciate the information. And just to let you know, so my son and we are participants of the Simons study. We've been part of the Spark study.
Thank you.
The question is, it says autism and neuroscience. Now that over 800 genes have been associated with autism, what do you think about epigenetics, neuroepigenetics, and research on autism?
Yeah, I mean, I do. That's one of the reasons. So, and first of all, I really apologize if I was over your head. So I apologize for that. So, but one of the new technologies called long read whole genome sequencing allows you to look at some of the epigenetic marks, looking at methylation on the DNA, and so I do think we'll learn something from that. Without a question. And so I'm excited. It's expensive right now. And so that's why we're trying to ask people, where should we be looking at this? So that's one way to do it. And I think that, I am kind of excited. So it's both the challenge and an excitement. Some of the NIH autism, ADSI, what is that? What is it? Data Science Initiative, that they're also working in our SPARC population, and they're looking at more environmental factors, mostly focused on like geocoding and looking at air quality, some socioeconomic status. And so I'm interested in they'll come out of that, because we have a lot of genetic information, and then how do we put that together with that? And of course, doing it in a way that protects everybody's privacy is sort of top of mind. So that's a lot of the work that we're doing now to make sure that that happens. But yeah, I appreciate your question. Thank you.
I'm CJ Sadel, a genetics fellow here at UC Davis. My question is sort of a comment and wanting your expertise. I'm always feeling like we don't have enough databases of structural MRIs to do kind of computational analysis, machine learning pipeline. I could see a future where we have, for all of those SPARC single gene and copy number variant disorders, an associated 10 to several hundred MRIs that I could download as a researcher, and then basically I can use machine learning algorithms to parse apart all the different regions and determine a neurophenotype, so we can kind of drive the phenotype into neuroanatomy in a way that a radiologist could never really do with the naked eye. So that's kind of one piece. Building that database, I'd be happy to submit an RFA for that, because it's a little thing that I want to dedicate my life to. But in addition, if there's any sort of thoughts on a database for neuroinflammatory axis or the pathways that are involved in neuroinflammation as they drive developmental disability and autism.
I mean, I think that's part of why we have that new ideas RFA, is because we want to hear different ideas that, things that we could address in a way that's like rigorous and that is scalable in some ways. I mean, I don't know that much about how easy it would be to build a big structural MRI database. So I would want to know that, right? I would want to know, like, can we really do that? How much heterogeneity is there across? So those are some of the questions we'd want to know. I do think that, there's clearly in autism, but in all of neuroscience, a really increasing interest and appreciation for neuroimmune interactions. And so, yes, I think that that's a really ripe area for investigation. I think designing experiments that are, going to be incredibly telling is going to be the trick there. But I think absolutely there's an interest in that. No question.
Concerning these larger databases, when you're compiling them and trying to make sure that they're usable for, broad-scale research purposes, what do you think about the, current concerns regarding possible overdiagnosis of autism?
Yeah, so it's so we're actually working, and I think I skipped over this on a slide, but we're working right now on a 5-year renewal for SPARC. And one of the questions that we're working on with the, SPARC has 31 clinical sites. And so that we're working on with the clinical site PIs is asking them about how do we, what is a high confidence autism diagnosis? So I'm actually really interested in understanding, what do we do there? And clearly, like one of those papers that I showed you, I think it was both the Varun Warrior and also maybe the Eric Morrow, that, looking at different age of diagnosis, and I would say that in SPARC at least, and I'm not a clinical psychologist, so I'm like relying on my colleagues, that there's a lot more confidence in the diagnoses at younger ages than in our adult populations. And so, but I think it's a really important question. I don't think we want to exclude anybody who wants to participate. And so, maybe some of the more longitudinal behavioral phenotyping will help understand what specific sort of phenotypes different individuals have. But I do, it is clearly a question. There was a study that was done, I think by Brian O'Rourke at OHSU, where they looked at SPARC participants. I think they were all children, and then validated that and found a very high validation using ADOS. But it would be nice to replicate some of that in the other parts of the cohort. But I'd be really curious to learn from anybody what they think would be the best measure for having being high confidence diagnosis.
Not only differential diagnosis of autism, but the complexity of multiple neurodevelopmental disorders in addition to autism. I mean, they're not necessarily in addition, it's the way we slice it.
Yeah.
But a child, even if you hold something like IQ stable, one child will have some language, another child will have great difficulty learning. One child will have lots of sensory reactivities, another child won't have any. These additional symptoms, which may or may not be, many of them are not specific to autism, but they co-occur.
Yeah.
And I'm assuming they have the same genetic and biological complexity as does autism without those occurring conditions. And so when you think about like your genetics slide, if you're actually looking at loading for the genetics of the other conditions, and many, many clinicians don't even define them. It's kind of like, I've always felt autism is it's a great big bucket, and once the autism diagnosis is made, there isn't the same passion to diagnose additional neurodevelopmental disorders. Is this language delay part of the autistic delay? Is it part of the general developmental delay? Or is it something specific? And you can say that with almost every symptom that is not one of the two core social symptoms. And that brings in much more biological and environmental complexity. but to get to the questions about what makes autism.
Yeah.
This is part of, I think, of the huge increase in diagnosis, but I think that's really ought to be taken seriously. And I don't think many of the differential diagnostic procedures that we use in these very complex studies are focusing on defining what is part of the autism and what is an additional load. And we're going to miss a lot until we can be clearer about those things. We're going to miss a lot of additional phenotypes and definition of specific subgroups in a biological way.
Yeah, I mean, to me it's really interesting or telling that if you look at Searchlight, in any of those conditions that the family groups are there because their gene has been associated with autism, but you can join because you have a variant in that gene. And, often it's like a third meet criteria for autism. 2/3 don't. They have other, so that tells you that there's that. And I do think that there's been, I wouldn't call it confusion, but that, what are we studying? And actually for SFARI, it started out studying autism, and I think these genes have been identified, have, seizure ID. The mouse models, those are really the things that are replicated in those. So I think that what we're trying to really struggle exactly with that question is how do we really get at autism? So I'm 100% in your corner there. Yeah.
Thank you for that great overview, and thank you, Sally, for kind of leading on to the question that I was going to get into. Was you spoke about heterogeneity in the phenotype and kind of how to parse them into different phenotypes, and also mentioned about the monogenic genes as causes for autism. But like we're all saying here, it's almost like the elephant with the 7 blind men, each one describing an elephant as to this is how my elephant is. And I have our fellow sitting here saying, yeah, the brain MRI is the answer, but that truly is not, right? So it's more of how do we take a human, and sorry I didn't introduce myself, I'm a clinical geneticist and chief of genomics here. Mm-hmm. So every day we are giving these results to the families and then hand-waving about the phenotypic spectrum where one person could, you mentioned, could have seizures, intractable, non-intractable seizures, and then the dad could be sitting there saying, yeah, he got it from me, but here I am. I may be a little bit on the spectrum, but not really affected as much. So I was wondering, if you have thoughts on how would we look at the elephant as a whole with all of these data that you have?
Guy, you guys ask hard questions. I'm like, no, I know it's not. I mean, I think that, first of all, that's what makes it interesting, right, is that it's really hard to do. I mean, maybe I'm naive, but I really do think that, being able to better understand the human condition, I mean, I think there's still a lot more in genetics that we have to do. I feel like we haven't really looked at the non-coding space very much. We haven't really looked at epigenetics very much. But I do think that we don't there's a lot of behavior that we just don't capture, right? And I know I'm influenced by, work in mouse. I used to sit there and watch mice freeze as a measure of learning and use a little stopwatch to figure out how long they were freezing. And now you can have depth cameras that record every posture in the mouse, and then AI approaches to analyzing that data, and you can pull out things that you didn't detect. So there is part of me that is hopeful that something like that will allow us. I don't really love the idea of a world where we're all walking around. I wear an analog watch, but I don't love the idea that we're walking around taking track of every aspect of our behavior and our biology. But I think that we're probably missing something. So that, maybe that's just maybe I'm naive about it, but I think that that's an opportunity for starting to look at that. I do think, there are clearly going to be environmental I think it's been hard to look at some of those because it's such a large space to understand. But I did learn today about some nutritional things that I think could be really exciting. And so I think that we just have to keep really open minds about what autism is, what causes it, all of those aspects. So
I have an easy question for you.
Oh, thank you.
And then Aubyn's going to ask probably another hard one.
That's fine. It's fine.
My question is, first of all, thank you for sharing all the information about the resources, databases, and so on. It is a bit overwhelming for us if we're not very familiar with all those resources.
Yeah.
And I was wondering if the Simons Foundation has like any kind of, expert sort of counselor or somebody who can help, listen to what the researchers' questions are, and then help direct them to what the resources might be.
Yeah, so we do have, that's what that team, the SDBR, the SFARI data, I'm terrible at acronyms, SDB, and biospecimen repository team. They're the navigators. You go to them, they take the question, they help you find the place. We're working really hard. I'm actually really excited about pretty remarkable advances in the last month in Claude Code and other AI that allow us to do better data visualization. And so I think we're working hard on trying to have good data visualization tools for people that will make it much easier to do. But we do, in the meantime, have a navigation team that's there. So if you go to the website and look for SDBR, there's a website, an email, they get more emails than anybody else at the foundation because there are always people asking them for, how do I do this? How do I get to this? Do you have this data? Do you have these biospecimens? How could I get those biospecimens? That's their job. Yeah. All right. Hard question.
No, I can't ask you a hard genetics question.
No, that's all right.
We've already met. So, but thank you for the careful way in which you've been doing this work. My question is more about the community. This is going to shock everyone.
Yeah.
About the community partnerships, because we are launching an initiative here, and we've really asked all of our scientists to support community engagement, and our basic scientists have been right on board. But I'd love to hear some examples of how that has worked for you, and what kinds of things you're getting from the community that's helpful.
Well, I think I told you about one thing that I loved last year, and we're doing it again this year, which is for all of our SFARI investigators, we offered travel supplements for the PI and 1 or 2 trainees to go to a family meeting, and then our Searchlight team gave them presentations about how to talk with family members. If they wanted to do a presentation, they ran through the presentation with them. And I would say it was probably a lot more impactful for the basic scientists than it was for anybody at the family meeting, but it really changed I think the way that they thought about the work that they did. I think that in terms of what, I think your question, though, really, if I understood it, is more like what have community members brought that's changed the way that basic scientists. I think a lot of it, one of the more impactful things that Spark has done is ask participants what things matter to you, and then taking that information and going, we should focus on this, or we should. Part of the reason for doing the Simons sleep pilot was because we were told from participants that sleep is such a major issue, and so that's why we did that. So I think that's one way of informing the work that basic scientists do. But I think it's a hard, sometimes there's just a divide in the way that, in communication maybe in particular, across the basic scientists, participants in a group. I would say a couple other things. We have had panels, for example, where we have scientists who are also parents of kids with autism talk about sort of their experience, and I think that is sort of a bridge sometimes for the other basic scientists. And I was last week at the Lurie Autism Center opening, and they talked about a program they started that I really want to do at the Simons Foundation, which is that they run a course for their trainees, and it's I can't remember how many parts, it's like twice a month, 3 hours at a time, and they go from like autism genetics to genetic counseling to community engagement to like every aspect of research. And I thought, oh, that's like, and I was going, well, we should do that and have our SFARI investigators. I don't think we could have them go twice a month for 3 hours, but something like that, because I always feel that we don't. Our last SFARI investigator meeting, there was this really great panel about, subtyping. And I remember Kathy Lord was sitting in the back and like kept raising her hand, and I had to run and bring her the microphone because the people doing the work didn't understand how to measure IQ. And so, and I should have had her on the panel. I like sat there and went, oh, I didn't have a clinician on that panel. I had only geneticists and basic scientists. So I think it's just education, maybe? I don't know, education, yeah.
Thank you so much for a truly edifying multi-level talk. This question sort of circles around stuff Sally was talking about and stuff that you've been mentioning. And it has to do with linking this ecological neuroscience initiative with educating researchers about the way we build the world. And your video analysis initiative for autism and the way to actually think about the evolution of human behavior and the way we create our realities by the affordances of our bodies and our environments for cognition is not taught in graduate school in psychology. It's not taught in neuroscience, but it's a major thread in cognitive philosophy. And since out of all those 200 applications, you guys chose a 10-year commitment to essentially an activist perspective, what is the explicit agenda to take that philosophical view of what humans do and what organisms do, from E. coli up, and bring it into the training of the basic researchers so that when they think about phenotyping, they're thinking with the lens of that kind of a dynamic, embedded, and active view?
I haven't gotten there yet, but I really like that vision. I mean, I really like that vision. And I keep thinking, like, the little steps that we take are to make sure that when we have that ecological neuroscience, when we have those workshops, that we bring everybody from the SFARI team to those workshops.
Yep.
So they hear all of it. So it changes the way that they potentially think about, what areas of study to support. And so that's the first step. But I do think that there's a huge opportunity there. And it was a combination of, it's an older idea, right? Affordances. And I think it got kind of overtaken by reward theory, the kind of classic computation, sensory and compute, motor out. And so it's an interesting, I just think I'm glad we're doing it. Also happened to be an amazing group of people who came together with a very cohesive vision about how to do this. And incorporated theory into it and experiments. So it was kind of the combination of a cool idea and a great structure for doing it. But I think it's, how do we start? They are going to be writing, I don't know where it's going to come out, because the journals decide that, but a piece, I think it may come out in Neuron, but that's about sort of the vision of that collaboration. So that's another way, is like telling the story about what they're trying to do, what their vision is for this. So that's another way. And I do think the transmitter, I don't know how many of you read the transmitter, but we try to get them to tell stories about some of these. And I think that's a pretty neuroscience community participatory thing. So I think some of it could go through that. But I like that vision, so thank you. I think it's a good one.
The UC Davis Mind Institute was founded in 1998 with the promise to reduce and prevent the disabilities that can be associated with autism and other neurodevelopmental conditions. Every day, our clinicians and researchers make progress on that promise. Our groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other conditions associated with disability are helping affected individuals achieve their fullest potential. Please visit our website or our social media platforms to find out more about current studies, upcoming events, and how you can help make a difference.
The preceding presentation is provided for informational purposes only. The views, contents, and opinions herein are solely those of the speaker and do not necessarily reflect those of the UC Davis Mind Institute or the University of California. This broadcast is intended for personal, non-commercial use. Do not make a copy, rebroadcast, re-encode, or otherwise distribute this broadcast without prior written permission from the UC Davis Mind Institute. Copyright 2026, Regents of University of California, all rights reserved.